Okay, everybody, this is the presentation on organic molecules. In class, you should already have done the packet with some partners called biological molecules, so you've already been introduced to this material. That will help me go faster. You also should have picked up a note sheet so that while I'm talking, you can take notes. If at any time you need to pause because I'm going too fast, you can always pause the screencast. All right, the first set of biological or organic molecules that we're going to talk about are the carbohydrates. Carbohydrates are produced by plants during photosynthesis, and they include only three elements: carbon, hydrogen, and oxygen. There are three categories of carbohydrates. The first category we're going to talk about are the monosaccharides. Monosaccharides are the simple or single sugars. They have a single ring, and that'll make sense in a moment. They're quick energy for us, and they have carbon, hydrogen, and oxygen in a 1 to 2 to 1 ratio. Notice glucose has 6 carbons, 12 hydrogens, 6 oxygens, 1 to 2 to 1. What's interesting is fructose has the same chemical formula, and so does galactose. But on top of that, what's interesting is that they have different structural formulas. So glucose, even though it has 6 carbons and 12 hydrogens and 6 oxygens, it actually is shaped differently than fructose. So when these two sugars, for example, touch your tongue, they taste slightly different. They're both sweet, they both are sugars, but the flavor is actually discernible because they have slightly different shapes. Okay, the last set of molecules were called the monosaccharides, and they were called monosaccharides because they were made of a single ring. The next category of carbohydrates are called the disaccharides. The prefix di means two. These are two single sugars that are put together to make a double sugar. They're also a great source of energy, and they are easily broken down into simple sugars, so they're easily digested. Examples of the disaccharides: maltose is a disaccharide. It is made of a combination of two glucose molecules put together. And that's what you see at the bottom of the page. Two glucose molecules, if you put them together in a dehydration synthesis reaction, which, if you remember from the packet, means you removed water. If you take water away, the two glucose molecules bond together and they form a molecule of a disaccharide called maltose. Sucrose is similar. It's a disaccharide. Instead of being two glucose molecules put together, it's a glucose and a fructose. Lactose, which many of you are familiar with, lactose intolerance. Lactose is the primary sugar that's in milk. And lactose is the joining of a glucose and a galactose molecule. Some people are unable to break apart the glucose and galactose monosaccharides from each other, and so they have difficulty digesting lactose. Okay, the last category of the carbohydrates are the polysaccharides. So we had monosaccharides, disaccharides, now we have polysaccharides. Poly means 3 or more, so it's 3 or more of those single sugars that are all bonded together. They get bonded together when we remove water, and you can actually see that in the diagram below. These are storage energies. They're more complex, and so they take somewhat longer to digest. You might get polysaccharides in foods like spaghetti. That they take a little bit longer to digest. Examples: starch, that's the diagram that's down below. Starch comes from plants and it's found in leaves and in tubers. Glycogen is made by animals and it's found in the liver and the muscles. And then cellulose is found in plants and it makes up cell walls. We're going to talk a little bit more about cellulose because it's kind of an interesting molecule. What I want you to see below is starch, though. Starch is nothing more than a whole bunch of glucose molecules put together. So 3 or more glucose molecules put together. So you see the reaction: 2 glucose molecules on the left, we're removing a molecule of water, and they bond together, and it's one after the next. Okay, like starch, cellulose is a polysaccharide. It's several glucose molecules put together. The primary difference between the structure, however, if you look carefully at the glucose molecules that have been put together to make cellulose, every other one is flipped upside down. It's a slightly different structure, and that is really important. Cellulose is the major building component of plant cell walls. It is a long chain of glucose molecules, so it should be a great source of energy. So why don't we eat leaves? Why don't most animals eat leaves? Humans can't digest it because we're missing the necessary enzyme to break those molecules apart into the simple sugars. We have it for starch when the glucose molecules are all face up, But when they flip upside down like this, our enzymes cannot pull those molecules apart, so we're not capable of digesting it. There is a protist in, for example, animals like cows. It lives in the gut of a cow, and it does have the enzyme necessary to break that molecule up. So a cow can eat grass. It swallows the grass into one of its many stomachs, and those protists help it pull the glucose molecules apart. So that's why a cow can get energy from eating grass. But humans, because we don't have that protist in our stomachs, and we don't have the enzyme, we can't really get energy from cellulose, so we don't digest it well. Okay, the next large category of biological or organic molecules are the proteins. So we're no longer talking about carbohydrates. This is a totally different category. The proteins, as many of you already know, are made up of amino acids, long chains of amino acids, sometimes thousands of amino acids. They are very important structural components of cells. They are found in enzymes, hormones, hemoglobin. Proteins are really important. What we're going to talk about primarily are the amino acids, and when you just put a couple of amino acids together. Proteins are such large molecules that we're not going to be able to look at them in much detail. Okay, so the building blocks of all proteins are amino acids. You join one amino acid to another in a dehydration synthesis reaction. You're removing water. Every amino acid has something in common and something that makes it different. First of all, in the amino acid structure, there's an amine group, and that's the area that's boxed in blue on this picture, the nitrogen with the 3 hydrogens. That's called an amine group, and every amino acid has it, and that's where it gets its name, from the amine group. That's where the amino acid comes from. Every amino acid has a hydrogen that's highlighted in pink. Every amino acid has a carboxyl or carboxylic acid group, and that's the one that's highlighted in green. So where does the name amino acid come from? Amino is the amine group, acid is the carboxylic or carboxyl acid group. And then finally, it has another group called the R group. R is not an element. There's no R on the periodic table. R stands for anything that's variable. This is the part of every amino acid. There are 20 of them. This is the part of every amino acid that's slightly different. We'll show you on the next page what some of those different amino acids look like. Okay, these are the 20 amino acids that humans use, and almost all living things on this planet use 20 amino acids. They look all very different, but you need to stare at them for a moment to see the similarities. Each one of the amino acids has a blue, it says H3N. That's the amine group, the N and 3 H's. Every amino acid has the green carbon with the 2 oxygens. That's the carboxyl acid group. And every one of them has a black hydrogen at the top. The part that varies from amino acid to amino acid is the section that's in red. If you're lucky enough to go on in biochemistry in college, you'll get to memorize this entire table like I did. Ooh, something to look forward to. Okay, now to build a protein, we need to join 2 amino acids together. You notice in the diagram that there's a highlighted area, an O and 2 H's. If you put an O and 2 H's together, you get water. So this is called a dehydration synthesis. When you're dehydrated, you've lost water. So we're pulling water away from these 2 amino acids, and we're causing them to form a bond with each other. And that bond is called a peptide bond. Now remember that we said proteins are hundreds or thousands of amino acids long. I can't ever draw that for you. So what we're going to look at are things called peptides, dipeptides, or polypeptides, which are basically baby proteins. I'll never be able to actually show you a real protein in terms of its structural formula. Several amino acids together make a polypeptide, and a protein obviously contains hundreds or more of amino acids. All right, the last major category of biological or organic molecules are the lipids. We're no longer talking about proteins or carbs. The lipids include our fats and oils. They contain carbon and hydrogen just like the carbohydrates did, but they have fewer oxygens than the carbohydrates did. What's interesting about the lipids is that they have twice the energy per gram than carbs do. So then lots of times students wonder, well, why isn't it good for us to eat lipids? We're supposed to stay away from fats and oils. Why isn't it good to eat lipids if they have so much energy? The issue is that they have too much energy. We don't burn that many calories in a day, and so the fats and lipids, the fats start to store in our bodies, and that's where obesity comes in. The lipids are used as long-term storage energy in animals. They are more difficult to, the energy is more difficult to access than the energy stored in carbohydrates, so we access it more slowly. Examples, phospholipids, steroids, and carotenoids. I'm not going to ask you about steroids or carotenoids, but I will teach you about phospholipids right at the end. Okay, lipids are identified by the presence of things called fatty acids. Fatty acids are what you see in red here. Fatty acids are humongously long, sometimes carbon chains with as many as 12 to 38 carbons in a row, and they have a carboxyl group attached at one end. So if you see at the very beginning of the red a C double bonded to an O, that's a carboxyl acid group. So remember we saw that in the amino acids they had a carboxyl acid group, and that's where the name amino acid comes from. Well, the fatty acids also have a carboxyl acid group. The carboxyl end is polar. That makes them hydrophilic. The prefix hydro means water, and phil means love. Just like the city, like Philadelphia is the city of brotherly love. Hydrophilic means that the carboxyl end is attracted to water. The hydrocarbon chains, the rest of that red area, is nonpolar, and it's considered hydrophobic. And maybe you can figure out, it translated literally, it means water-fearing. So it doesn't like to be near water. So the fatty acids are not easily brought into water. They're repelled by water. Now, a triglyceride is what this is actually a picture of. The word glyceride comes from the word glycerol. The part of the molecule that's in green is a glycerol molecule. That is an alcohol. It is not a lipid, but when three fatty acids attach to that green glycerol molecule, we call that a three fatty acids, triglyceride. So it says it's three molecules of fatty acids attached to one molecule of glycerol. All right, many of you have heard of saturated and unsaturated fats. We talked a little bit about it in the biological molecules packet that you did in class. Saturated fatty acids have no double bonds between the carbons. Now be careful, you do see a double bond in red between a carbon and an oxygen. That is always there, and it doesn't count. When we're trying to determine if something is saturated or unsaturated, we're looking for double bonds between carbons, not between a carbon and an oxygen. In general, the saturated fatty acids are unhealthy. They're usually solids at room temperature, and they include the animal fats like butter. Okay, the unsaturated fatty acids are much healthier. They have at least one double bond, which causes the molecule to have a bend in it. And you can see that in the lower molecule. They are healthier in general, except for the trans fats, which are the worst of all. We learned in the packet today that trans fats are normally liquids at room temperature, but then humans hydrogenate them. They add hydrogens, which breaks the double bond and makes them solid at room temperature so that they're easier to bake with. But it turns out that that also makes them much, much less healthy when we purposely hydrogenate them. So most of the time, the unsaturated fatty acids are liquid at room temperature, except the trans fats, which have been hydrogenated and they become solids at room temperature. Substituting unsaturated for saturated reduces cholesterol levels. Okay, this is the last slide. Woohoo! The last category of lipids that I want everyone to be aware of are the phospholipids. Phospholipids have a polar head, and then they have two fatty acids attached to glycerol, not three. Remember, the triglyceride had three fatty acids. The phospholipids have two fatty acids. On the right, you see a yellow ball with two blue tails coming out of it. It almost looks like a person, like a 2-year-old would draw a human being. Those two blue things coming down, those are the fatty acids, and then the head, this is a polar head, and we'll talk about that in just a moment. Cell membranes are made of phospholipids, 2 layers of phospholipids, and that's what you see in the black box down at the bottom right. You've got 2 layers of phospholipids. The legs, or the fatty acid tails, point in toward each other, and the heads, the yellow balls, stay outside from each other. The reason is that the head is hydrophilic. That means, again, that it's water loving. There is water on the outside of a cell membrane, and there's water on the inside. I've got to say that. There's water on the outside of a cell. I have to say that right. And there's water on the inside of a cell. There's no water in the cell membrane itself. Okay, so the heads tend to point toward the water on the outside or toward the water on the inside of the cell. The tails are hydrophobic. They're made of lipids, they're fatty acids. They don't like to be near water, so they automatically orient themselves inward into the cell membrane. And that's it.